Good morning. It's September 30th, and this is your daily brief in science. Here's everything you need to know. A recent study highlights that nearly all cardiovascular events are preceded by identifiable and controllable risk factors, emphasizing the importance of prevention and early intervention. The study analyzed health records spanning over 10 years and found that more than 93% of patients had 2 or more risk factors, with hypertension being the most common. In the United States, over 93% of these patients had high blood pressure, while in South Korea, the figure was 95%. Even women under 60, often considered lower risk, showed that over 95% had at least one non-optimal risk factor before experiencing a heart event. The findings suggest that managing blood pressure, cholesterol, blood sugar, and smoking is essential for reducing cardiovascular risk. Lifestyle factors such as adequate sleep, exercise, healthy nutrition, stress reduction, and maintaining a healthy weight are also crucial. The study notes that even mild elevations in traditional risk factors are significant and should be addressed through lifestyle changes or medications. It also points out that using higher treatment thresholds, like 140/90 millimeters of mercury for blood pressure, can miss many at-risk individuals who have levels above optimal but below these cutoffs. There exists a clear gap between optimal and clinical thresholds for risk factors, with many individuals falling into non-optimal ranges that still pose significant risks. The data underscores that managing these risk factors is feasible and vital for prevention. Yet current healthcare often focuses more on treatment after events occur rather than on early risk management. A new AI tool developed by Harvard Medical School aims to improve the diagnosis of brain tumors during surgery. The system, called Picture, can differentiate between glioblastoma and primary central nervous system lymphoma, two cancers that often look similar under the microscope. Picture achieved over 98% accuracy in distinguishing these tumors, which can help reduce diagnostic errors in real time. It uses advanced pathology image analysis and includes an uncertainty detector to flag cases where its confidence is low, prompting human review. Designed for intraoperative use, Picture provides immediate insights that can influence treatment decisions, such as whether to remove the tumor or pursue radiation or chemotherapy. The AI was tested across 5 international hospitals with more than 2,100 pathology slides and is supported by the NIH. It outperforms both human pathologists and other AI models, particularly in difficult cases. The model is publicly available to support further research and aims to democratize access to neuropathology expertise. Most samples tested were from white patients, indicating a need for validation across diverse populations. Future updates may expand its capabilities to other tumor types and include genetic data. Its consistent performance across different tissue types suggests broad clinical potential, and ongoing development aims to improve diagnostic scope and accuracy. Researchers at the University of Tokyo and Riken have announced a breakthrough in synthetic biology, a method to produce all 21 types of transfer RNA simultaneously in vitro. This development addresses a key obstacle in creating self-replicating molecular systems. Previously, scientists had succeeded in reproducing the 20 amino-acyl tRNA synthetases necessary for protein synthesis, but lacked a way to generate all tRNA types together. The new approach encodes all tRNA genes on a single DNA plasmid, transcribes them collectively, and then processes them into mature molecules using HDV ribozyme and RNase-P enzymes. This streamlines the production process and eliminates the need for individual handling. The achievement paves the way for constructing complete, self-sustaining protein synthesis systems outside living cells. Such synthetic systems could surpass natural organisms in stability, scalability, and customizability, with potential applications in pharmaceuticals, industrial enzymes, and bespoke biomolecules. The platform is expected to evolve into a versatile, cell-like environment for studying molecular biology and biofabrication. It also enables the incorporation of non-standard amino acids into proteins, expanding possibilities in protein engineering. This milestone supports efforts to build life-like molecular machines capable of self-reproduction, offering insights into the origins of life and evolution. The research was published in Nature Communications on August 26, 2025, under the title Simultaneous in Vitro Expression of Minimal 21 Transfer RNAs by tRNA Array Method. Chinese scientists have announced a major breakthrough in superconducting magnet technology. On September 28, 2025, researchers at the Institute of Plasma Physics, Chinese Academy of Sciences, developed an all-superconducting magnet that generated a steady magnetic field of 35.1 Tesla. This sets a new world record, making it roughly 700,000 times stronger than Earth's magnetic field and about the same multiple more powerful than natural magnetic flux. The magnet was built using high-temperature superconducting insert coil technology nested within low-temperature superconducting magnets, allowing it to achieve high field strength while maintaining operational stability. During testing, the magnet was energized to 35.1 Tesla, operated stably for 30 minutes, and was safely demagnetized, demonstrating its reliability. Although it is not yet integrated into a fusion reactor, this development advances the platform for experiments at such high magnetic fields, which are vital for future fusion research. The team addressed previous challenges related to stress, shielding currents, and multi-field effects, enhancing the magnet's stability under extreme conditions. This breakthrough has potential implications for multiple fields, including aerospace propulsion, power transmission, MRI technology, nuclear fusion, and magnetic levitation systems. It also supports the development of superconducting instruments like nuclear magnetic resonance spectrometers and contributes to efforts in fusion energy and space propulsion. To put this into perspective, Earth's magnetic field is about 0.00005 Tesla, and MRI machines operate around 3 Tesla, making this new magnet strength truly extraordinary. Achieving superconductivity at such high magnetic fields requires extremely low temperatures, which remains a challenge, especially in fusion applications where heat from reactions complicates maintaining superconducting states. This achievement aligns with China's broader fusion research initiatives, including participation in the ITER project, which aims to build the world's largest fusion reactor. Recent research from Chinese scientists has provided new insights into the molecular evolution of type 5 CRISPR-Cas systems with a focus on CAs12, a key genome editing tool. The study emphasizes that the diversification of RNAs derived from transposons played a significant role in this evolution, highlighting RNA-level innovations rather than changes in protein structure. Findings show that RNA innovations enabled TNPB proteins to become CRISPR-like systems capable of guiding DNA targeting. This discovery opens new possibilities for engineering smaller and more versatile genome editing tools. The research also examined how transposon activity, involving TNPB nucleases and their relation to CAs12 effectors, contributed to the emergence of CRISPR immunity systems. Experiments demonstrated that artificially splitting TNPB's rRNA into 2 parts could convert it into a CRISPR-like system that utilizes guide RNAs from CRISPR arrays. Further engineering confirmed that this split rRNA approach was enough to transform TNPB into a functional CRISPR-like system. Genomic analysis identified 146 TNPB-like proteins and uncovered 6 intermediate clades called trans-Cs, which represent evolutionary stages between TNPB nucleases and CAs12. Phylogenetic and structural studies, including AlphaFold modeling, mapped these 6 trans-C clades as key intermediates in the transition from transposon systems to CRISPR immunity. The research traced the lineage of these proteins, illustrating their importance in CRISPR system development. It also found that some trans systems use dual-guide RNAs, both intrinsic CRISPR RNAs and transposon-derived rRNAs, indicating their role as evolutionary intermediates. This dual-guide RNA mechanism marks an evolutionary step towards the complex guide systems seen in modern CRISPR-Cas systems, demonstrating the sophistication of these intermediate systems in facilitating DNA targeting. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.